it's time to start. And it's obviously giving us plenty of pleasure to bring Kevin Booth, who is in Michigan right now, although we were hoping to have him in Tampa today, who's an associate professor in the Department of Biophysics at the University of Michigan. Kevin is both an experimentalist and a theoretician. He got a bachelor's in chemical physics from Center College, master's in biology from UC San Diego, also a PhD in theoretical and mathematical physics from UC San Diego. He then moved to Harvard, where he spent 6 years working in molecular physics and systems biology before he, about 10 years ago, was recruited by the University of Michigan as faculty member in the Department of Biophysics. So he has been working in theoretical and mathematical models of evolution in microbiomes and drug resistance and evolution and how to treat microbial populations in more efficient and more evolutionary enlightened ways. So there is a lot of parallels between what he does there and what we're trying to accomplish here at Moffitt. And obviously we thought it would be great to have him here to give a talk. We'll be more than happy to at least have him on soon and bring him over here in Tampa on other states. So Kevin, the stage is yours.
Okay, thank you very much. I appreciate the invitation. I really wish I was there in person. I was really excited about it. I had a trip planned to go visit my parents afterwards, who live not too far away. I was really excited. Fortunately, I got sick, so I've had a little fever and I've been feeling great the last couple of days. But I'm really glad I still get to talk to you. Might have to lean away for a cough or sneeze from time to time, but it shouldn't spread through the screen, so I think we're safe. So I work in my lab along with my lab on bacteria, but we're somewhat organism agnostic. So I think a lot of the concepts that we deal with and the way we think about problems would apply to any self-dividing organism, if you want. So hopefully you'll see some connections. I've for a long time seen connections and been inspired by the work done there, and so it's really cool to get to talk to you. I hope we'll get to talk more. I will tell you, we work typically on this bacteria called Enterococcus faecalis, or at least for the things I'm going to talk about today. Now, this is a picture that I took right when I got to Michigan. We got our microscope and I took it. I think this is the only piece of data that I took with my hands. And you might think the image looks blurry, but that's definitely your screen because it's a beautiful image on my screen. So these are bacteria involved in things like endocarditis, infections of your gums, urinary tract, bloodstream infections. Again, the specific biology is not going to come into play a whole lot. We tend to look at things at an ecological level. And so we're not going to talk a great deal about that. The types of questions we ask, if you want to drill down to the simplest explanation, we have a population of these cells and we add a drug that's supposed to kill or slow the growth of these cells, antibiotic. We want to know what happens. And in the simplest view, you could say, well, maybe one of two things happen. The population is going to survive or it's going to die. And our research is often motivated by the fact that this survival thing is happening more than we'd like it to, more than we'd like it to in medical communities. And so this is a hard problem, potentially global problem and a long-term problem potentially, what's known as antibiotic resistance. So of course there's analogous problems with treatments for viruses and cancers. And so the main goal of our lab is trying to understand evolution in these communities and see if we can control it in some way, if we can shape the communities by understanding it and maybe manipulating the environment that they live in. So in order to do that, there's some essential information you have to know. So in particular in my field, historically, it's been very important to know what exactly is happening at the molecular level. We zoom in, on a cell. We can characterize what's causing that cell to be resistant. There's been a tremendous amount of progress in this field over the last, 3-4 decades. And I tend to think of the mechanisms that cause resistance as sort of one of four main classes. This doesn't cover everything, but it's close. They are either molecular mechanisms that keep the drug from getting into the cell, so they decrease the uptake of the drug. Mechanisms like efflux pumps that pump the drug out of the cell once it gets in. So in either way, you're lowering the intracellular concentration. Cells might make an enzyme that degrades the drug. So in that case, that could be either intracellular or extracellularly. The concentration of drug literally and physically is reduced. Or you could have some type of target modification. Now, I know in cancer, there are a lot of sort of more complex systems-wide mechanisms of resistance. In bacteria, these are typically the way we think about it. And these mechanisms are, of course, essential, but what I and a lot of people in this field looking at the problem the way we do are trying to argue is that this information is completely essential, right? It's important to know what's happening, but it's incomplete. And it's incomplete because bacteria don't live by themselves, right? They're not single cells isolated from the environment. They live in these dense and often heterogeneous communities where there's lots of cells. So something like this, maybe in today's day and age, is how we might think of a community. And so the question that I like to think about, or that we're trying to think about, is essentially, does having neighbors matter, right? So the fact that you're not living alone as a cell, does that matter? Does your interactions with the environment and with your neighbors, does that change the behavior of the community? And this is a conceptual problem that arises often in physics, also in other fields, but since physics is the field I come from, and it happens in particular in statistical physics, which is the field that I was trained in. And in that situation, you have these microscopic interactions. These are basically just small scale interactions. In physics, they're often at the atomic level, so interactions between individual atoms. And there's these suites of mathematical tools that include things like dynamical systems theory and stochastic processes, and generally what I call statistical physics, which encompasses some of these, but of course doesn't. But of course, this has been developed in other fields as well. And these tools let you go from this microscopic behavior, these small scale interactions, to macroscopic behavior, some larger scale description of the system. So the classic example in physics is magnetic dipoles, where the individual atoms interact in a particular way. And when you get many, many of these atoms together, the behavior of the system is governed by statistical properties that describe, say, the sticky behavior of magnets, right? Ferromagnetism. And so we do something similar. We think about problems in a similar way. We take molecular or phenotypic scale information about resistance, often information that's gleaned from thinking about things, at this sort of drilled down scale. We try to develop coarse grained models. We don't include everything. We try to sort of develop, essentially minimalist models. They're often stochastic and nonlinear. And we use those models to make hypotheses that we can test in the experiment or in simulations. And the main question that we try to ask is, can we exploit this knowledge that we learn about sort of how these small scale interactions give rise to larger scale population behavior? Can we exploit this for better control of a population? So just one example of the type of experiments that we do. I think several of the experiments I'm going to talk about today deal with this. We often do very simple experiments. So we and many others have developed bioreactors where you can simulate infections, right? And so what I mean by a bioreactor is just basically a little test tube here. The tube has at the top silicone tubing that comes in and out of the top of it, sits in a little cup holder that we manufactured at the machine shop. So very, very simple. Each of these tubes is connected to a pump. That pump can pump out liquid into or out of the vial, and all this is controlled by a computer. So there's lots of wires, and it looks, the way a physics lab should look sometimes. So, and we can do things like simulate particular dosing schemes, right? Say, try to periodically dose a drug over time. Or we can do things like adaptive therapies, where we measure the population at a given time point, and then we decide what to do based on that measurement or based on some historical measurement. So they're very simple systems. I think right now, each one of these is about 18. We have several of them that we've made now. So we can do, 30 or 40 vials at a time. So it's not high throughput, but it's not zero. And then on the side of these, just drill a hole. We have a little light on one side, a detector on the other. These are just infrared light. So we flash light, and we essentially see how much of that light gets through the solution, or in other words, how cloudy the solution is, how much light scattering there is. And you can measure the voltage from these little simple $1 detectors, and they correlate really well with optical density you can measure from a spectrophotometer that's much more expensive. So it's a really simple way of monitoring sort of cloudiness of the solution, which is proportional, at least in some regimes, to the number of cells there, the colony forming units, it's often called in microbiology. And so we can get growth curves, just the population density over time. And for example, they'll look exponential over some range, like you'd expect it bacterial populations and reach some carrying capacity. So we can do this for each file. And so, these are sort of the tools we use, a combination of math models. We also use microscopy. I don't think I'm going to talk directly about any of that today, to look at more, three-dimensional colonies of bacteria. But so one of the first questions that we've been thinking about for a while is, can we use cell interactions to control resistance, the emergence of resistance? And so this is a collaboration I did with several researchers in evolutionary biology. Andrew Reed, he's worked a lot with different approaches to evolving pathogens. Robert Woods, he's a medical doctor here at Michigan, also an evolutionary biologist. Jason Karslake, he's a PhD student in my group and also was a postdoc, now a professor at Penn State. And so the question we wanted to ask is, can we use cell interactions to control resistance? And what's the simplest type of interaction you might expect? Well, it's competition, right? So if I start really simple, I have a mixture of sensitive cells drawn in blue here, resistant cells in red. They live in some, green patch of nutrients. For our purposes here, sensitive cells are inhibited by the drug. Resistant cells are not inhibited by the drug. So, simple toy model situation. And as the cells grow, the nutrients get depleted. And as a result, that can affect the growth of the other cells in the community. So very, very simple, well-known type of interaction. And we actually modeled this in this case with classic Lotka-Volterra competition equations. So these are really simple nonlinear differential equations that involve a linearly dependent growth rate term that depends on the population density. So they're simple and well understood equations where we just tweaked them to match this situation. And the idea here that we wanted to test was whether or not it was possible in these well-mixed communities. So these are just stirred in a test tube. Is it possible to use drug to control the sensitive cells, right? Because they do respond to the drug. And then by doing so in a judicious way, control the resistant cells and do that through the fact that sensitive cells deplete resources and can potentially slow their growth. And if you could do this type of thing, and this has been addressed quite a bit in cancer literature, for example, if you could do this type of thing, then what that would mean is that you want to use the drug wisely, right? You don't want to load the drug up at the very beginning necessarily, because what could happen if you use a lot of drug, you get rid of your competitively suppressing sensitive cells and you allow the resistant cells to grow unimpeded. So at least it's possible that there could be cases where the presence of these sensitive cells, because they are controllable by the drug, would be beneficial. And so we did a simple experiment. So we have these vials that I told you about. We have populations of resistant cells. In this case, they're E. coli, but we've actually done this with other species too. You have resistant cells that are E. coli. You have sensitive cells, also E. coli, so red and blue, just to distinguish the two types. We're going to measure the cell density in real time, and then based on what we measure, we can adjust the dose of drug that we apply to the vial. And we're going to do that with the goal of keeping the population below some threshold density for as long as we can. So just a really simple feedback algorithm that says, the density's too high, add more drug. The density's not too high, sit back and wait. And so the hope is that you'll do this by using as little drug as possible. And again, the idea is that we want to keep these sensitive cells still growing somewhat so that they are still using resources and therefore potentially affecting the resistant cells. And then we just measure the escape time. So that's the time when our population eclipses some threshold, which we define up front. And then we're also going to do two control experiments for comparison. We're going to do the exact same experiment, but in sensitive only populations, and the exact same experiment in resistant only populations. And so the difference in, say, this top vial and this bottom vial is just the presence of the resistant cells at the same ratios, or at the same total densities. And similarly, the difference between the top and the resistant only vial is just the presence of sensitive cells at, again, the same initial densities. And so all three of these are going to get the exact same drug schedule. So we're using the mixed population to decide on that drug schedule because we don't know up front, what's the best schedule to use. So we're just going to do it sort of adaptively, try to use as little drug as possible. But whatever we decide to use, we're going to apply it to all three populations. So the experimental data looks something like this. So this is the population size over time. I've drawn a dashed line here. This is our arbitrary threshold. We call it P max. So we can set this however we want. If growth eclipses this threshold, we're going to call that treatment failure or escape time, right? And so, so the data looks like this. So in the sensitive only populations, we're able to keep them below the threshold density. That's not particularly surprising. We've added only sensitive cells, and the timescale of these experiments are such that de novo mutations in this population is not super common, right? So we don't expect to see that on these timescales. So essentially, the sensitive cells that we put in remain sensitive for the time course of the experiment. But when you do the same experiment with resistant cells, of course, you can't control them, right? So even though we add more and more and more drug, there's no way to keep them below this dashed line forever. And so we would call this time the escape time. Okay? And what's cool is when we do the same experiment in the mixed populations, it turns out that even though the resistant and the mixed populations have the exact same starting numbers of resistant cells, the ones that we can't control, we can keep the mixed populations below this threshold for the whole length of the experiment. And again, the only difference is the fact that one of these populations has sensitive cells in it. So that was exciting, kind of cool. It suggests that that this controllability does have an effect, and it has an effect even in populations in a test tube like this that are well mixed. There's no spatial component. There's no local competition because of that spatial component. So it's somewhat surprising to us to see such a strong effect in this type of well mixed system.
Can we ask a couple of questions?
Sure.
So I'm intrigued about, first of all, what's the mode of action of the drug or of the treatment? And then is the proportion of sensitive cells a specific factor here?
Yeah, two good questions. So the drug is a protein synthesis inhibitor. It's a tetracycline derivative, doxycycline. There is not, to our knowledge, any cooperation between the sensitive and resistant cells that's known. So it's the resistant cell has a mutation in the target of doxycycline. So at least, to our knowledge, there's not any strong cooperation. We didn't see evidence of that in the experiments. The ratio of initial resistant cells does matter. The general trends hold. In other words, they're easier to control, but both the ratio matters. And then another thing that matters, but that we don't see in our experiments, is the mutation rate. Right. So if you had a population of sensitive cells with, say, a mutator strain, had a higher population or higher mutation rates, it's an experiment we're trying to do now, you would expect that, this benefit provided by sensitive cells would decrease substantially and could reach a crossover point where adding more sensitive cells is actually bad. Why? Because not just do they provide more, overall bacterial burden, but they also provide a reservoir for new mutants.
Right.
And it just so happens in this case on these timescales, and for many timescales with bacteria, based on calculations of the model, we wouldn't expect to see the mutation rates matter for typical mutation rates in this experiment.
So I'm looking at that figure now. I see that there's a kind of bump in the sensitive, but it seems to match some sort of dynamic that's going on with the drug.
Exactly.
So that is some artifact of the way the drug was delivered or something.
Exactly. And the model, because both the model and the experiment start from, not perfectly decided initial conditions in the model, we randomize some part of it. So the drug dosing is not the same in every experiment. What you're seeing here is sort of an average. But yes, exactly. The up and down of the sensitive cells is just an artifact of the specific drug treatment.
So it's not a delay in death.
It's not a delay. So we did measure a delay time in when we started to see the population growth slow down. So in a simple experiment, just sensitive cells, you add drug to them, you wait and see how long before the population starts to slow down. I want to say it was around 30 minutes. We included that in the model for quantitative reasons. It didn't provide much qualitative difference, but it did, for example, at the beginning here with the sensitive cells, they grow, upwards at the beginning, even though we added drug early on.
Yeah.
That's basically the delay time.
Cool. Thanks.
Sure. Okay, so what you might expect here is that this competition is going to depend on the cell density. It's going to depend on, how many bacteria can you tolerate? So in our test tubes, we don't care, but of course in an infection or something, this would be an important parameter. And so we could ask, well, what happens if this threshold density, this allowable amount of bacteria, decreases? And so first, I'll just show you a simple way of quantifying the escape time. So this plot just shows the time to escape, the time when we eclipse threshold. And this x-axis is really just two situations. It's going to be this high density situation that I've showed you. So this is just quantifying the data you just saw. So the mixed populations we can hold in all but one experiment for almost the length of the experiment. The resistant only populations escape much earlier, about a little less than half the time. So you can quantify this, this difference. This would be your escape time delay, if you want, due to the presence of sensitive cells. And so the question we went on to ask next is, what happens if we lower this threshold, right? So I showed you one with a relatively high threshold. What if we bring our threshold down to here? So we can only tolerate a small total population of bacteria. And so you would expect that in that case, competition is decreased, the average spacing between cells is decreased. And essentially, if the density gets low enough, different cells shouldn't feel the presence of one another. And we see that. Right? So when we lower the threshold, we do the exact same experiment. Now the escape times are very close to one another. So the take-home message is that, sure, sensitive cells can prolong containment, but only if you can tolerate a sufficiently high density of total cells, a sort of total population size. And so we did this for a lot of different population sizes. So this is an example of what the data looks like. This is just the cell density, so the population size, volume's constant here, so I use density and size interchangeably. This is the cell density over time. For each of these curves, we're trying to control them at one particular density. That density is given by the color of the curve. So blue is lower thresholds. We're trying to stop them earlier. Red is higher thresholds. And then I cut the curve off once they crossed threshold. Right. So each color is a different value, but it becomes, hard to see, more transparent once you've crossed threshold. So for example, this curve was growing fine until it reached its threshold, which is right here. Right. So beyond that, they don't show the curve anymore. And so you can even see by eye that when you use populations that are only resistant cells versus mixed populations, that there's a big qualitative difference, right? The resistant only cells, the threshold escape time, is clustered in this region, roughly, 6 to 10 hours or so. Whereas in the mixed population case, you see a sort of trend heading towards, up and to the right, if you want. So lower thresholds tend to escape earlier, larger thresholds tend to escape later. And you can quantify this and compare it to the model. So this is your escape time or failure time as a function of threshold density. And so this dashed line, this is our model with a mixed population. The dashed line at the bottom is the resistant only population from the model. And the marks are experiments. So the squares are the mixed population experiments. The X's are the resistant only. And so it does a surprisingly good job, I think, given the noise in the experiments, the fact that this is biology happening in a lab. The relationship was remarkably good, I thought, or the agreement was remarkably good. Again, this is a simple model of just essentially Lotka-Volterra competition with this adaptive treatment built in. One thing that's interesting in the model, and this is what I was getting at the previous slide that we saw, once you pass a particular critical value of Pmax, this sort of cusp right here, the escape times are the same. And so there is some critical value of the density where it becomes beneficial to add these sensitive cells, and you take off towards the upper curve if you're a mixed population, or the lower curve if you're resistant only. Prior to that density, competition isn't sufficiently high to help you.
Can I ask another question, Kevin? So I'm not quite understanding. So the threshold density, are you changing that by changing the nutrient supply or something? Like, how is that? Good
question. No, that is essentially just programmed into the algorithm. And it says when you get to this density, you need to add enough drug to keep you below that density. So it's an arbitrarily selected threshold.
Right. Okay.
Yeah. And we do the experiment so that we're always below, as far below as we can keep it, we're below the actual carrying capacity of the population. So it would grow higher with no drug. But then, we take a subset of that below carrying capacity and we decide where we want to try to hold them.
Because I'm sure you're aware of some of the adaptive therapy literature. One of the things that we've looked at and what's been shown is that as you get close to carrying capacity, the competition obviously intensifies, right? So is that something that you looked at?
Yeah. So that's captured, in a sense, by the separation between these two curves. Right. So the fact that, when we have larger and larger threshold, we're getting closer to carrying capacity.
Right. Okay.
So, we see this large separation. Now, we stay away from carrying capacity, completely carrying capacity in bacteria, partially because they go into stationary phase and their physiology changes quite a bit. So
no, that makes sense. It's the same in cancer cells too.
Okay.
You can't really get to that experimentally. It gets messy. But another point in your Lotte Volterra model, you only showed it briefly, but was it just a straight competition term, or did you have something a bit more sophisticated in there?
No, just a competition term. Yeah, okay. Competition, no interactions. Otherwise, competition is equal to between the cells.
Oh, really? Okay.
Yeah. So it's the boring case mathematically where there's like a line of fixed points.
But it works.
Yeah, right. So we can't solve it analytically with this adaptive term in there, but yeah, it was cool that such a simple model works. Okay, so the take-home message, if you want to contain a population of resistant cells, at least under these conditions, one thing you could do is add controllable cells. And the idea is that, the drug controls the sensitive cells, and the sensitive cells indirectly through resource depletion can control the resistant cells. Okay, so that's one thing we did. We're trying, a lot of sort of offshoots of this. We see cases, it looks like preliminarily in other species with higher mutation rates, cases where this will backfire on you. Adding these more sensitive cells, as I said, provides a reservoir of resistance also, or potential resistance. So you can see cases where it can backfire. And so we're trying to sort of get to the bottom of that now. Now, another sort of related but slightly different approach would be to use more than one drug, right? So this is a commonly studied idea, certainly in cancer and also in antibiotics. What if you cycled the drug over time? So you use drug one, then you switch to another drug, maybe you cycle it in some periodic way or some random way. Wouldn't that help slow down resistance? So this has been studied, for decades, very well studied problem. Now, at least in bacteria, the sort of party line here is that this should not necessarily help reduce resistance. So cycling is often not helpful, or at least it's less helpful than using the drugs together, at least. And so, that's sort of the standard thought about cycling. But there's one thing that's been left out of a lot of these, at least until about 7 or 8 years ago. And that's that, as you all know from cancer, that resistance to different drugs can be correlated. And so just as a cartoon plot, if I'm considering 2 drugs and the resistance to drug 1 increases, then the resistance to drug 2, some unseen drug, some drug that these cells haven't been exposed to yet, might also increase. And that could happen, for example, in the most obvious case, if the 2 drugs are very similar chemically. For example, there's an efflux pump maybe that pumps out one of these drugs, it could pump out both of them. So we call this cross resistance, or some people call it collateral resistance. But there's also, the other side to this coin, which is that increasing resistance to one drug could be associated with decreased resistance to a second and potentially unseen drug, something called collateral sensitivity. And so in bacteria, as far as I can find, this was first reported in 1952. And I think this has been a topic that's been looked at under different names quite a bit in cancer, much less so in bacteria until about 10 years ago. So relatively little work on this collateral sensitivity idea. But around 2013, Morton Summers' lab did this very innovative work where they tried to use this collateral sensitivity to slow resistance. And the idea was, if you have, say, a pair of drugs, and resistance to drug A gives you sensitivity to drug B and vice versa, then you could potentially use a cycle of those 2 drugs to slow resistance. And you could do this even with larger cycles. You could develop these sort of networks where you create this beneficial collection or cycle. And since then, there's been many, many papers on this in the last decade trying to understand this. But there are still a lot of open questions. These are increasingly answered. At least when we started working on this, some of the big questions were, are these collateral effects common, or is this a sort of very rare phenomenon involving very specific types of drugs? Are they repeatable? Can we use them then to design an optimal drug sequence? So is it really periodic? Can we do better than that? And how would we even think about that problem in a systematic way? So we started with experiments just trying to characterize this in this particular bacteria. So we just use lab evolution to evolve cells to increasing concentrations of drug each day. So we start with a population of what I'll call drug sensitive cells, add them to a population, and then each day dilute into fresh media with increasing amounts of drug. So this is work from Jeff Maltus, a PhD student in my lab, who actually now is a postdoc with Jake Scott, which I think many of you know. So at the end of this experiment, we isolate one of these mutants and we can measure, say, a dose response curve. So just the population size as a function of drug. So this just characterize the resistance of that mutant to the drug, in this case, pegocycline. It's a protein synthesis protein synthesis inhibitor antibiotic. And we can characterize this resistance in a number of ways. We usually use this IC50. It's the half maximal inhibitory concentration. So reduces this curve down to just a number that tells us how resistant the cells are to that drug. But then instead of just measuring to the drug that we expose the cells to, we also measure this response to a library of drugs, in this case, 17 different drugs. So now we have this sort of resistance to each of 17 drugs when you've been selected by one particular drug. And now we repeat this whole experimental process for all 17 drugs. So now instead of selecting with drug 1, we perform the evolution in the presence of drug 2, and then drug 3, and then drug 4. And for each of those, we measure our response characteristics, our resistance to each of the drugs. And so you end up with a matrix that looks something like this. So this is on a log scale, but essentially the blue spots in the matrix correspond to collateral resistance. These are cases where the isolated mutants have higher resistance to the drug in question than the ancestor strain. And the red is collateral sensitivity, which is the opposite. So on the x-axis, this is the drug we used for selection. So they're listed here if you're interested. I'm just numbering them here. So this is the drug used in the evolution experiment. On the y-axis, this is the drug we used to test it, to characterize it. So this is the resistance we're looking for. So just as an example, if I use drug 14 in the experiment and I isolate a mutant and I want to then find out what's the resistance of this mutant to some testing drug, I can just look in this column, right? So for example, if I wanted to say, what's the resistance to drug 14, the one I used for evolution, well, of course, it's blue, it's resistant, right? Otherwise, it wouldn't have lived through the experiment, right? So the diagonal of this type of matrix is always going to be blue. There can be different shades of blue. For one reason or another, evolution in the lab works better or not so well, depending on the specific strain and drug. But all of them evolve some level of resistance. And then, something that's maybe a little more surprising was that the off-diagonal elements are also frequently non-zero and pretty large. So there can be a change of 4x or more in the effective concentration needed to kill these drugs, the MIC or something equivalent. And we see both blue and red. We see both resistance and collateral sensitivity, relatively frequently, right? It's not a super rare occurrence. And so one of the first things we wanted to ask is, how repeatable is this? So instead of doing this with one population, what if we do it with 4 populations each, right? So now our matrix just gets a little bigger. We can zoom in and get a sense of what's happening. So this is a case where we selected with daptomycin. It's a cell wall targeting antibiotic. So we see that all 4 of the replicates are blue. That means they've evolved collateral resistance, or resistance in this case. That's not surprising, right? Daptomycin was used to select for these cells. If they didn't become resistant to daptomycin, there would have been no experiment. But you can see that some of the off-diagonal elements, for example, the response to ceftriaxone can be really variable depending on the population. So in this case, we had one population that became very resistant to this second drug, one population that became very sensitive to it, one that essentially stayed at the same level of resistance as the ancestor, and then another that became very resistant to it. And we've actually zoomed in on this particular situation quite a bit recently. We're about to submit a small paper where we understand a fair amount about where this variability has come from in this particular case. But on a larger scale, the question we wanted to ask is, can we use the correlation structure in these profiles, right? Can we use these matrices, if you want, that nature hands us to design a better drug cycle, right? So we consider sort of this matrix as a set of constraints that's, imposed by nature, right? These trade-offs are there. They're set by perhaps the specific species you're looking at, the specific drugs you choose for the matrix. But if I'm handed this matrix, if I know this relationship, how in principle would I even design a drug cycle? And if I design that cycle, would it actually work? Is there actually a way of slowing resistance by choosing drugs in a particular way over time? So we modeled this using what's called a Markov decision process. Many of you may be familiar with this. It's a relatively simple extension of a Markov, just typical Markov chain. The idea is we have a discrete time process, and we're going to define the state of the system at each time step as just a vector that contains the profile, the sensitivity profile to each of our drugs. So this is just, you can think of it as just a vector that's got a number in each of these spots. That number tells us whether we're sensitive to the drug or more resistant to the drug than the ancestor. And the idea of this type of process is at each step, we decide what drug to use. So this is called an action. So we get to pick one of our 17 drugs and we get to apply that at that time step. And then based on what we apply, we're going to roll a dice. There's some probabilistic stochastic process that determines the sensitivity profile at the next step. So there's this, conditional probability, this transition matrix, if you want, that's going to determine this process. We inferred this from evolutionary data in the simplest way possible, which is essentially, we said that sensitivity profiles add up linearly. So if you apply the same drug over and over and over, you become increasingly resistant to that drug. In other words, you would get, say the blue stripe, the blue portion of this, it gets higher and higher and higher over time. These are on a log scale, so that additivity comes down to like multiplicative effects in terms of actually looking at IC50s. So this was the very simplest way we could think of to do it based on the somewhat limited data that we had. And so, you could imagine all different evolutionary dynamics that you could bake in here, much more complicated potentially. And so, okay, so the next question is, how do you choose the drugs then? So you derive what's called a policy, an optimal policy. It's just a mapping from your space of profiles, so your sensitivity profile, your resistance level to each drug, and it maps to an optimal drug. It tells you what drug to use based on what your sensitivity profile is. And the goal, which you can choose for any particular problem, but for us was to optimize the sensitivity to the applied drug in some time averaged way. And so specifically what we wanted to do is minimize this term RC. It's looks complicated, but it's really not. This is the instantaneous resistance to the applied drug. These brackets just mean it's an expectation value. It's a stochastic process. So we're taking essentially an average. And then you have this parameter here. It's called a discount factor. So gamma is just a parameter that ranges between 0 and 1. So it's just a scalar parameter. But we multiply it by t, which is the time step. And what this is saying is that as this term gets closer to 0, what does that mean? It means that we're maximizing the drug efficacy at the very next step. So we call this like an instant gratification policy, right? It's saying that which terms show up here, only the most recent ones, right? Only the ones with t the smallest, right? But as gamma approaches 1, it means that you keep terms farther and farther out in the future. So you don't just care about what happens right now, you care about what happens down the road as well. And so you can tune this parameter to see either, instant gratification, quote, treatments, or treatments that might have some longer term efficacy. So you can change this time scale. And then we just sum over an infinite time horizon. This is a sort of technical detail that doesn't matter. The good news is this type of problem is not hard to solve. It's been solved with dynamic programming. Hard to get analytical results depending on the situation, but numerically it's quite easy to solve. And once you have the policy, once you know this mapping from drug profile to drug of choice, once you have that mapping, this just reduces to a standard Markov chain. And so it's very easy to analyze. So here's an example of what you might see. So we limited it to just 6 drugs in this example. And we said, well, what would a policy predict? How should you optimally treat some population based on the experimental data we measured, this matrix? And you can see what it predicts, and this is fairly common, is it predicts these sort of almost periodic cycles, but with some weird aperiodic things thrown in, right? So maybe you almost cycle back and forth between, say, rifampicin and fosfomycin for a little while, but then you throw different hydrocycline at this step, ampicillin at this step. So first we just asked numerically, how do we expect this to perform in a model? And so we took, again, just this sample of 6 drugs, we reduced it to this Markov chain, and we just simulated it. And so first, as just a comparison, we took the matrix and we stripped out any collateral effects. So in this case, it's a diagonal matrix. The diagonal elements mean that you become resistant to the drug you're selected with, the drug you see, but there's no effect on any other drugs. And so the model, as you would expect, just increases slowly. It doesn't really matter how you choose your drugs. You can do worse, but you can't do better than just approaching resistance, very quickly. So this is the resistance to the applied drug on average. It's over many trials. It's a stochastic process over time. We put an artificial limit on this. So the ceiling here is artificial. So it would just keep going up. So resistance in this case is going to quickly reach the highest level that it can get to, even if you optimally apply the drugs. And that's not surprising. The drugs are essentially independent here. But if we add back in the collateral effects, you could first try by just randomly cycling through the drugs. And for this particular matrix, a random cycle would give you something like this. So you improve on the no collateral effects case, right? But you still reach a high level of resistance relatively quickly. And this spacing between sort of the maximum resistance and where this random settles essentially is going to depend on what these off diagonal elements look like. How likely are you to be sensitive versus resistant when you randomly choose a second drug? So that is matrix specific, although most of the matrices look something like this. We're not seeing matrices that are, say, very, very sparse, for example. So still a steady march to resistance, but on average, we haven't hit the maximum resistance. So, maybe a little improvement. Now you could consider this instant gratification policy. So this is an optimal policy, but it's targeted at treatments early on. And you can see early on, the level of resistance actually goes down. So you actually sensitize the populations early on, but eventually you hit a U-turn and that doesn't work so well. And over long time periods, it does even worse than the random case. Well, what was cool, we thought, was, what happens as you choose this gamma to be close to 1? You see if you can maintain low levels or minimize resistance over longer time scales. And in the model, somewhat interestingly, you can maintain low levels of resistance indefinitely. Now, we don't expect that to happen biologically because this matrix itself should be changing over time on some time scale. But it's somewhat interesting that, given the actual matrices we see, the ones nature hands to us, it's at least possible over the time scale where these matrices are valid, where collateral effects are described by a matrix like this, to choose drugs that hold resistance considerably lower for, indefinite periods of time. And so, you can fine tune what you want out of this treatment by changing this parameter, by choosing this time scale. Do I need, instant gratification? Do I need a really strong, effective treatment now? Or do I need something that's going to last over longer times? And we can ask a little more about how it's working because it's such a simple model. So we could say, for example, should we always use highly effective drugs? And by those, I mean drugs that tend to produce low levels of resistance when we apply them. And what about less effective drugs? What about drugs that, our experiments suggest, lead to resistance pretty quickly? Should we ever use those? And because it's such a simple model, you can calculate the distribution of how you should use the 2 drugs. So this is the probability of the steady state probability distribution for how to use drugs based on the resistance level you'd expect them to generate. And this is direct resistance to that drug. And you see that it's strongly bimodal in this long-term solution. So what this is saying is most of the time, the largest fraction of the distribution, you choose drugs that work really well. In other words, drugs that are highly effective today. They generate low levels of resistance. And that makes sense, right? The idea here is, you're trying to constrain the pathogen growth now. But you sometimes sprinkle in at a lower frequency drugs that do promote resistance, right? At some lower frequency. And the idea here is that these are steering the population towards a more vulnerable future state. They're not as effective today, but they're allowing evolution that could potentially weaken the long-term viability of the population. And so we think of this optimal policy as a balancing act. You can use less effective drugs or more effective drugs. The more effective ones help you today. The less effective ones potentially help you longer term by steering you towards a more vulnerable future state. And we tested this experimentally. We used just 4 drugs. This is already a nightmare of an experiment to do. We did 4 to 10 replicate populations in one of 4 conditions. One was just single drugs. So we used each of the drugs on their own. So ampicillin, and it would be ampicillin every day, day 1, day 2, day 3, day 4. We also looked at all 2 drug cycles. So drug 1, 2, 1, 2, 1, 2, 2, 3, 2, 3, all the different 2 drug cycles. And then we looked at a uniform 4 drug cycle, 1, 2, 3, 4, 1, 2, 3, 4. And then we compared it to the optimal policies. And we measured growth cumulatively over time in these populations. And we did this for about 20 days. So, the advantage of bacteria is they grow very quickly, right? So we can get quite a few generations in 20 days. So this is what the cumulative growth looks like. Each of these, first, this dashed line is just growth without drug. So this is on an exponential scale. It's not clear what the units are in this graph, but it's effectively an exponential scale. So small differences are actually quite big. Each of these curves, light curves correspond to individual replicates, dark curves correspond to averages over these replicates. So these are replicate populations. And so each one of these average curves corresponds to one of the 4 drugs, right? So in this particular example, resistance to tigecycline seemed to appear, relatively quickly. Or said another way, the total growth in tigecycline-only treatments was the highest. And we have similar results for 2 drug cycles or an all-drug cycle. And the take-home message here is just that different drugs are different on these time scales. One thing that's interesting is the 2 drug cycle that performed the best is a cycle that is of the type recommended in some of the original work on collateral sensitivity in bacteria, where it's 2 mutually collaterally sensitivity inducing drugs. So resistance to drug 1 tends to cause sensitivity to drug 2. Resistance to drug 2 tends to cause sensitivity to drug 1. And so that's consistent with what you might expect intuitively. But of course, different pairs will perform differently. And then an all drug cycle, on this particular experiment, under these conditions, tends to perform, as well as anything else. And then we compared it to the optimal policy, and it outperformed everything, pretty dramatically. And almost every single replicate outperformed every single other replicate. So in other words, every replicate of the optimal policy outperformed even the best performing replicates from the others. So, strikingly clear results, especially given the complexity of this process and the simplicity of this model. So we were really excited about that.
Could I ask a couple of questions? That's
sure,
very cool, Kevin. This is definitely an area that we're interested in, and maybe not quite as in depth as you've got into it, but the idea of kind of an evolutionary double bind is something that we've been thinking about with sequences of drugs, right? And can we exploit that? Unfortunately, there is multi-drug resistance is a very common outcome. I don't know if that's a common thing that you see in bacteria, but it's definitely common in cancer cells.
Absolutely, yeah.
Yeah, it is. And so that double bind strategy, does it, this is specific, this idea of it being kind of low sensitivity versus sort of a high sensitivity, and then or low resistance versus high resistance. And, the sequencing of that. It's like, do you take a risk with that high resistance, but it actually potentially makes your future sensitivity even higher?
That's exactly, yep.
Yeah, and so, that's something that we think could be very applicable in cancer. But one of the big problems, of course, is that there's a sort of dynamic to how those drugs are delivered and wash out. And so we can sort of theorize about sort of cool sequences, but how practical are they? And so I'm intrigued about, how do you actually sequence those drugs in such a rapid way and get it washed out and do all of that? How does that actually practically work?
Like in the clinic?
No, I mean in these experiments.
Oh, in our experiment, yeah. So in our experiments, it's not hard to do because each day we dilute the population into a fresh environment, and we pick the drug in that environment. So we are completely neglecting the potential dynamics of drug degradation. It's there the entire day. Except what the cells might do to degrade it, but there's no outside influence. There's no background, degradation or dilution rate. And then each day we have a new environment. So
it's kind of almost like a replating equivalent.
Exactly. It's exactly equivalent to that, just in test tubes or these bioreactors. Now we have done experiments not with this many drugs and not so complicated, sort of trying to merge these things, where we do consider different dynamics, but it's typically been with even just a single drug, and there we focused more on how different cells interact in those. So for example, cooperative resistance. So we could in principle look even experimentally at more complicated dosing regimes. This was actually, in this experiment, this was actually done in well plates. So it wasn't even with the bioreactors I showed you before. So it's just diluting each day into new environments.
The thing about replating that we've seen is that there's a risk that you're selecting for the winners each time, which is maybe what you want, but it could also be not what you want if you're thinking about collateral future effects, right?
Exactly. Yeah. So I think, are you saying like essentially a bottleneck effect that's selected? Yeah. And I think that's a super interesting thing to look at. We thought about looking at it and just, haven't yet, is how these bottlenecks could change it, because, on the time scale so we switched drug every 1 or 2 days. So for bacteria, that ended up being, in these experiments, something like 20 generations. So we would switch every 20 generations. And at least what we've seen in separate experiments, we're getting populations that are fairly well dominated by one strain at that point. So it's like you said, we are essentially selecting for a winner, we think. Yeah. And that's not necessarily, the right regime to be in all cases.
Yeah, exactly. But that's the thing, is in sort of similar computational approaches, not to this, but we're using sort of agent-based models, we've seen that if we to do something that's more of we actually just let them sit in the dish, we don't replay, and we play out some of these scenarios. Now, it's in silico, we can do that because there's not all that toxic crap happens, right? You get a totally different trajectory.
Really? I can imagine that could happen. That's really interesting.
Yeah. But some we can talk about another time. But
because certainly, in these systems, we can, and of course with models, but also in these experiments, we can probably mimic that in different ways by changing the bottleneck or by changing, doing them in these bioreactors. So,
sweet.
So I'll leave. I'll stop relatively soon, but a word of caution, these are in vitro experiments. This is of course not anything close to a patient. Even though we have a lot of them, this is not a hospital. Right. And I think probably the most important thing is I'm not a real doctor. I'm a physicist playing around with models of evolution that I think are super interesting. So we are collaborating with people here, trying to apply some of that. For example, this project, we're trying to think about how we could apply this to recurrent urinary tract infections in spinal cord patients, where they get these recurrent infections and therefore they have these effective cycles of drugs, but the way the drugs are chosen is somewhat arbitrary and often leads to a bad outcome where you become systematically resistant one drug at a time until you run out of drugs. So we're trying to design both retrospective studies and then even prospective studies with some clinicians here at Michigan. But all the work I've told you about today, is purely in vitro. So it's very important to keep that in mind. Controlling evolution, really hard, as you all know, and I think it's super interesting on its own. So we start really simple. We control the conditions very well. It's already non-trivial. It's interesting in very simple mathematical models, even without many of the complexities of biology that we know exist. So we think it's kind of cool that at least in these in vitro systems, given these oversimplifications, that we're still able to make some progress at least. I'll just mention other things that we're thinking about that would probably interest you. We've also looked at problems that involve combinations of drugs. So this is just a plot where you see growth as a function of concentration of two different drugs. We've done evolution experiments where we pick a contour of constant growth. So, roughly a contour where growth is the same. So where the combined effect of the drugs, at least in terms of growth rate, is the same. And we evolve them at all these different places. And you, of course, can see quite different outcomes. We can understand this to sort of a 0th order approximation using some rescaling arguments, which I think are kind of interesting. We've also looked at cooperative resistance. In this case, for us, it's been resistance facilitated by resistant cells producing an enzyme. That enzyme, even though it stays within the cell, it's not excreted out into the space, but it makes the cells effectively sinks for drug and can reduce the effective concentration. So we've looked at, we see interesting effects over time. For example, cases where increasing the total population size, depending on how you do it, can actually promote population collapse. And then in space, things get really interesting, and we really don't know what's going on. So this is an example of an experiment where, as you go up, ampicillin, the drug is increased. As you go to the right, we change the initial fraction of resistant cells. These are individual colonies on a plate. Red is sensitive cells and green are resistant. The cells are engineered to express different proteins, so they look different colors. We see lots of different cool effects here. We don't understand them all. We have one or two papers on this, but it's an ongoing thing that's quite complicated, as you all know. Spatial behaviors matter a lot. We even see very different spatial behaviors depending on the scale we look at. So if we look from far away, we tend to see what often looks like large patches, cells of like cells clustering together. If we zoom in to a single cell level, though, we see enrichment and spatial structure even on the scale of a few microns. So there's interesting stuff going on here. We're trying to understand how these two sort of almost opposite effects can happen as you change scales. We're also using these simple flow reactors to try to build in multiple habitats, so like islands of cells, where say you can fix the drug concentration at a different value at each island, right? So in this case, 3 habitats. We set it up so that we manage flow between those habitats, and we can call this flow, quote, migration if we want, and we can control exactly how migration changes between the different habitats. And again, you won't be surprised to know that this heterogeneity, I'm calling spatial heterogeneity, it's really just 3 islands, so it's not a continuous space, but this heterogeneity can really change the evolutionary dynamics. So this is the growth of a population over time. And so this sort of rapid increase in growth that happens at around 30 hours, that's a resistant subpopulation taking over. And that changes a lot depending on how you set up the concentration in these vials. So both of these and several related projects are coming soon. So we can certainly talk more about that. I didn't quite have it yet in presentation ready, pretty graphs, but it's interesting stuff that I think you guys are thinking about too. So you can read our papers. We also do some non-bacterial work and got a tiny bit every now and then in statistical physics, and some other stuff. We're collaborating with some cancer biologists actually doing cell migration stuff. It also turns out my brother is a cancer biologist. I think he has actually visited even Chris Wood from Duke. So he takes a quite different approach than this, but it's interesting that when we started grad school, we went in like very different ways. I wanted to do only theory, and he became more and more interested in cancer biology. But we met up again on this sort of resistance problem, so it's interesting, I think. Most of this work's ongoing in some form. Got lots and lots of unanswered questions, so love to chat, email, Twitter, or hopefully in person someday soon. So Thank you again for the chance to talk to you.
Thanks. That was great, Kevin.
Yes, it was great. This is, I guess, time for questions. And I know that Sandy will have some. He has already expressed some questions and comments during the talk, but it's probably a good time to let other people, if they have any questions, to use the time now. Well, while I prepare, I guess that one of the questions I have is that I don't have experience working with bacteria myself, but one of the things that I don't know if that applies to your model system, but there are cells that are not tumor cells that actually also contribute to the resistance from treatment size. They impact the treatment. So, for instance, working with Andrei Marusik, who is also in the audience, we know that fibroblasts secrete factors that allow for the tumor cells to resist treatment.
Yeah.
Is there anything quite like that?
Oh, you froze. I think I got the whole question though. So there are surely, immune related factors or other things in real in vivo systems. But there's not an obvious analogy that jumps out to me though, like that. Cause yeah, I mean, I know the fibroblast connection and those sort of external signals. And I know from I think both you all and from Jake's group, we've talked about this as a game, a evolutionary game, which becomes really cool. I don't know the analogy right away. I wonder about it. There's lots of sort of black, dark matter in this in these microbes, and especially biofilms, and they're secreting all these factors, and they're forming these sort of complex extracellular matrices that have DNA as part of it, and it's not super clear what's going on there. So probably, but not that we know of. Let me say that.
So another comment from someone who is more used to cancer systems, the treatment, the drugs that you're using are really sort of something you can sort of assimilate to some of the treatment options in cancer clinics. But obviously, one of the things that people have been working on for the last, I don't know how long over here, is immunotherapies. Isn't there anything that you can use to sort of prime the immune system to target bacterial cells? And if that's the case, do they cooperate to evade the immune system?
That's a great question. So I don't know that area, the specific immune side of it. So I'm not particularly familiar with the work in that. I will tell you in a very, very simplified way, we have started to look at that question a little bit by doing the following. We grow bacteria in our vials. So we have this whole time series of their dynamics. And then we've used math models of immune response that are based on, so it's usually coupled ODEs, where one of the ODEs is the pathogen population. Well, we're actually measuring that, but we can feed it into the model, numerically solve for the activation of the immune system. And then when the immune system is active, we turn on the pumps and pump cells out. And so it is not at all a biologically valid approximation, but it's a sort of mechanical analog where we can, and in those systems, we can start to see things like, if you add the drug at the wrong time, you see these they're essentially like predator-prey cycles, where the goes up and then the pumps come on, and then so you see this like limit cycles and things like that. So I guess the short answer is we're not so much looking at, the real immune system in that response. I haven't followed that super closely. We are starting to think more generally about environmental responses that depend on the history of the bacterial population, not just in an adaptive therapy way, but like, can we sort of mimic some type of immune dynamics? And if so, what happens to the evolution? Like, do they learn to evolve to these particular, temporal dynamics? I don't know the answer yet.
Very cool. Anyone else has questions? Sandy, you have any more questions yourself?
Of course. I was waiting for others.
I wasn't afraid.
Oh, there's one from Goja.
Oh, cool.
Okay, so it says, I have a question.
Oh, let me see, yes. Can you read it? Or
in cancer, we can have tolerant cells that give rise to resistance through adaptation.
Do you know tolerant cells in cancer?
Oh, so by tolerant cells, are these, they're not genetically different? They're like, it's like a phenotypic response. It's is that what that means?
So and actually one of the things I'm getting over my Zoom is that Sandy, I don't know if you can make Andre a participant. Andre is an experimentalist and he's been working on this tolerant cells, so he'll have a different definition than me. But tolerant cells don't thrive in the context of treatment, but they don't die, or at least they don't die as much as normal cells.
So yes.
I don't see Andre there. Andre
is an attendee right now.
Oh, sorry. Okay.
Yes.
Yeah. So in bacteria, we call these persister cells.
Okay. And yeah. I'm confident it's the same thing.
Or I think, if I understand correctly, that's the analogy.
Yes.
Can you hear me?
I think I hear you now, Gos. Yeah.
Okay. Excellent. So yeah, in the context of bacteria and persister cells / tolerant cells, in your model, I don't think I have seen them, right? I think like your experimental setup was either just fully resistant cells / sensitive cells, and I'm just wondering if you can comment on like how realistic is it in the real life bacterial system. By the way, this is super cool work. I really liked it.
Oh, thank you. You're asking a great question. How realistic is it? I don't know, because I know there are these persister cells. One thing that limits us in the lab, well, I think of it as a positive too, but to really to measure dynamics of cells, we have to stay at a drug concentration that doesn't just wipe everything out. But of course, in clinical situations, you often want to use considerably higher drug concentrations than this. And so it could be that, these persister cells play more of a role there, where populations get completely, wiped out, except for these dormant, subpopulations that then sort of reseed the population later. We can do experiments like that, where, we use much higher concentrations, and persisters have been a topic of a lot of interest over the last, decade or so in microbiology too. We haven't really focused on them, but I would say they are important, but it's hard, it's really hard for me to say how realistic or not we are by ignoring those.
Great, fantastic stuff. Really cool.
I think I've met you maybe once before.
Yeah, we've met over Zoom. With
nice
Jake
to see you again.
And I was actually the one who invited your brother to give a seminar.
Oh, okay. Oh, sorry about that. No, he's my little brother, so I'm always.
So the question is, I'm somewhat related to Gosha's question, but like maybe more agnostic of. So what if you start with naive cells? So to get resistance, you have to passage them through increasing concentrations. But what have you explored when you start with the therapy naive populations to see what's schedule would be beneficial, and then it might enable playing with more clinically realistic dosing schedules where you cannot take like a little bit of a different antibiotic pill every couple of hours, but limited to kind of a week of this, a week of that, and then to the point of where we cannot give the concentrations of the doses that would wipe everything in clinics. Obviously, if they were wiping everything that they're using clinics, but if we're wiping everything in clinics, there wouldn't be a problem of antibiotic resistance. But oftentimes you have the issue of kind of accessibility, or if you have a biofilm or something, three-dimensional growth, and the bugs inside that might have less access to the antibiotic. And I see that you started kind of exploring some complex through environmental context, but have you thought about starting with naive cells and having it in the spatial context to find whether these inferences still hold? Because I think it would really, make
so we have, and we're starting along that exact path. There's there's so much to look at that we've left out here so far. But yes, that's absolutely one of the sort of the in general, the spatial. Well, we did everything I told you about today was also in these well-mixed test tubes, which is, not a realistic model of an infection, right? Except maybe particular, urinary tract, maybe, but, it's these biofilms that are three-dimensional structures. And so all of that, as you know, and I'm sure same in cancer, those are, I guess cancer, often you don't have confluent cells. So, but anyway, those are, of course, much harder experiments. So we're moving slowly on those, but yeah, we are thinking about it.
Well, then the cancer depends on how you do it. If you do it because the way we do it in petri dishes in 2D, it's not realistic as in tumors. They are confluent.
Oh, right. Okay, sure.
It's possible
That
to do them in 3D. It's just like more.
It's a pain. Yes, that third dimension is. I tell people in my lab, like, we're good with usually one dimension or infinite dimensions, but if you get in between there, it's a huge pain. You know?
Cool. Well, Kevin, thank you very much. This was
I've
great.
got a couple.
Oh, you're not okay. Sorry.
Sorry. So, you left that kind of juicy spatial stuff to the end just to sort of tantalize us, right? That's a bit unfair.
we don't know what we're doing yet completely.
But obviously the kind of tumor niche and the dialogue between the tumor and the non-tumor is a really kind of big focus area for most of IMO, to be honest, because that is obviously a dynamic feedback, right? And that sort of transformation that potentially occurs temporally is also happening spatially. One of the things that we've sort of been intrigued about is, resistance as driven by the niche versus resistance as driven by a mutation or some epigenetic change or something.
Oh, I see.
And so if you imagine all of that's probably happening simultaneously, unfortunately, and so having a cool experimental setup to play out some of those scenarios could be fun. But more specifically, one of the niches that we've been interested in is metabolism, a metabolic niche. And the fact that we know in cancers, especially some of those more resistant populations, may be gaining that advantage through a change in their metabolism. And it's always a bit of a chicken and the egg. Is the metabolic change because of their resistant behavior? Is the metabolic change facilitating their resistant behavior? Right. But do you see kind of metabolic changes in these populations? You never touched on that. I'm not sure if there is, but for example, for many cancers, they become very glycolytic, and as a result, they produce a lot of acid, and that acid resistance becomes a key element to their survival.
Oh, that's interesting.
Acidity hurts populations that are not acid resistant, and that could be other tumor cells, but it could also be non-tumor cells.
That is really interesting. We haven't looked much at metabolism. We do see if we do evolution experiments in these bioreactors, we do see, even in the absence of drug over these timescales, usually a couple of days, we will see an increase in growth rate, and you can see mutations that are associated with different carbon source usage or things like that. So it does look like they're adapting to the media, but we don't have not these complex feedback loops that you're talking about. But yeah, there's probably something there, but we haven't really looked at it. What's interesting is one of the very first papers we did with these vials is we grew cells at different fixed densities, and we held them at a fixed density by just pumping in more media when they got above that density. So it wasn't using drug to keep them at that density, it was using flow. But by using that flow, we could then infer the growth rate of the population. And so then we would take a fixed amount of drug, we put it everywhere, both in the vial, but also in the input vial. So drug's constant everywhere, and just looked at growth as a function of cell density, all below carrying capacity. So in bacteria, all in this exponential regime. And so the classic response, in bacteria would say, well, they're an exponential phase, they're all going to respond basically the same to drug, but they don't. And there was a big difference sometimes in the growth. And in many cases, we don't know the mechanism, but one of the things we thought of is that it could be a change in the acidity of the environment. And it turned out when you use strong buffer, there were a few drugs, I think it was 2 or 3 of them, where that density effect was entirely determined by the acidity of the media, the changing acidity of the media. And the other thing that was interesting is the effect went both ways. There were some drugs where the changing acidity increased the effectiveness of the drug. And so higher density populations actually were dying faster, and the opposite case. So there's certainly these feedbacks happening. We've sort of accidentally indirectly stumbled into it there, but that's most of it so far.
Yeah, no, that's very intriguing. That definitely sounds like there's some parallels there. I mean, obviously, you can't do everything, right? And, you know, we're all sort of attacking this problem of resistance from every angle we can think of as a group. But if there's ways to sort of feedback or synergize across these organisms, I think, you know, we should leverage those opportunities, right?
Absolutely. Yeah, absolutely. It would be, yeah, really, really cool, I think.
Thanks.
Okay. Once again, thanks, Kevin. This was really a thought-provoking talk, and
oh, thank you. Yeah.
Your ideas are something that jive very well with this is the work we do over here as well. So we're definitely looking forward to having you on stage maybe later on this year. Yeah,
I'm really looking forward to it. Thanks for the invitation and for letting me adapt when I needed to adapt. So.
Quite.
All right. Thank you again, and yeah, I hope we'll get to talk more soon.
Cool. Well, bye everyone.
Bye.